Hello everyone. Today we will discuss about important topics in biological oxidation. Long answer questions. First one, electron transport chain. Electron transport chain is a series of 5 complexes, that is 1 to 5, located in the inner mitochondrial membrane that couples redox reactions to produce ATP through oxidative phosphorylation. These complexes are complex 1, NADH coenzyme Q oxidoreductase or NADH dehydrogenase, complex 2 succinate dehydrogenase or succinate coenzyme Q oxidoreductase, complex 3 cytochrome BC oxidoreductase, complex 4 cytochrome oxidase, and complex 5 ATP synthase. Electrons from NADH and FADH2 are passed through these complexes and release energy to pump protons, that is H plus, into the intermembrane space to create a proton gradient. Here oxygen is the final electron acceptor and forms water. Second important question is enzymes involved in biological oxidation. All the enzymes which are participating in biological oxidation belong to the class of oxidoreductases. There are 4 subclasses: oxidases, then dehydrogenases. Dehydrogenases include aerobic and anaerobic dehydrogenases. Then third class is hydroperoxidases and oxygenases. Oxidases catalyze the elimination of hydrogen from the substrate, which is accepted by oxygen to form mostly water. For example, cytochrome oxidase. Second one is dehydrogenases. Dehydrogenases cannot utilize oxygen as a hydrogen acceptor. They catalyze the reversible transfer of hydrogen from one substrate to another and bring about oxidation reduction reaction. For example, NADH dehydrogenase, succinate dehydrogenase, etc. Coming to hydroperoxidases, hydrogen peroxide is the substrate for these enzymes. For example, peroxidase, catalase. Fourth one is oxygenases. Oxygenases catalyze the direct incorporation of oxygen into the substrate molecules. For example, monooxygenases, dioxygenases, etc. So these are the important long answer questions. Coming to short answer questions. First one is chemiosmotic hypothesis. This is also called as Mitchell hypothesis. This theory deals with the generation of ATP by ATP synthase. Chemiosmosis involves the pumping of protons through special channels in the mitochondrial membrane from inner to outer compartment. Second one is oxidative phosphorylation. Oxidative phosphorylation is the final stage of aerobic respiration occurring in the inner mitochondrial membrane. Here NADH and FADH2 are oxidized to produce ATP. It involves electron transport chain that pumps protons and creating a gradient used by this ATP synthase to phosphorylate ADP. Next question is uncouplers of oxidative phosphorylation. Two types are there: physiological uncouplers and chemical uncouplers. Physiological, for example, thermogenin, unconjugated bilirubin, long chain fatty acids, etc. Chemical, for example, 2,4-dinitrophenol, dinitrocresol, pentachlorophenol, valinomycin, aspirin, dicumarol, etc. Next one is inhibitors of electron transport chain. Complex 1, pyrisidin, rotenone, amobarbital, chlorpromazine. Complex 2, trienoyl trifluoroacetone or TTFA, then malonate and carboxyl. Complex 3, British anti-lewisite or dimecaprol, antimycin A, fenformin, naphthoquinone. Complex 4, carbon monoxide, azide, cyanide, and hydrogen sulfide. So these are the inhibitors of electron transport chain. Next question is inhibitors of oxidative phosphorylation. First one, atractyloside, which inhibits transport of ADP in and ATP out of the mitochondria. Second one, oligomycin, which binds with enzyme ATP synthase and blocks the proton channel. Third one, orovertin, which inhibits F1 subcomplex. Fourth one, venturicidin, which inhibits FO subcomplex. Next important question is malate aspartate shuttle. This shuttle transfers electrons from cytosolic NADH, which is produced in glycolysis, into the mitochondrial membrane. As the inner mitochondrial membrane is impermeable to NADH, this shuttle uses malate to transport electrons. Next one is glycerol 3-phosphate shuttle. This one transport electrons from cytosolic NADH, which is produced in glycolysis, into the mitochondria for oxidative phosphorylation, and here regenerates NAD plus to keep glycolysis running. Next one is high energy compounds. Molecules that release significant free energy upon hydrolysis, typically more than 7.3 kilocalories per mole. For example, nucleoside triphosphates like ATP, GTP, CTP, UTP, then phosphoenol pyruvate, 1,3-bisphosphoglycerate, acetyl-CoA, phosphocreatine, etc. Next important question is stages of oxidation. Biological oxidation is the cellular process of generating energy primarily through the oxidation of foodstuffs in 3 stages. Stage 1, primary metabolism, that is digestion and breakdown. Here, macromolecules like carbohydrates, proteins and fats, these are broken down into smaller subunits like glucose, amino acids and fatty acids. This is primary metabolism. And coming to stage 2, that is secondary metabolism. Here, production of reduced coenzymes like NADH, FADH2 through Krebs cycle. Then stage 3 or tertiary metabolism. This is the final stage, that is ATP generation through electron transport chain. It is also known as terminal oxidation or oxidative phosphorylation. Next important question is mitochondrial diseases. This include MELAS, that is mitochondrial encephalopathy with lactic acidosis and stroke-like episodes, NARP, that is neurogenic weakness, ataxia and retinitis pigmentosa, then Leigh syndrome, infantile ophthalmoplegia, then LHON, Leber's hereditary optic neuropathy, and MERRF, that is myoclonic epilepsy with ragged red fibers. So these are the important topics in biological oxidation. This is today's topic. Thank you for watching.